Hello, everyone. Today we're going to talk a little bit about the digestive system, which is another important system in the body whenever you're eating and such. But let's get into it then. So the digestive system processes what we ingest and absorb. So let's just write this. Digestive system. We have two functions, ingesting and absorbing. So ingesting is to basically, well, the main idea is you first have to chew it down and be broken down, which is digestion. Ingestion would be trying to get it to your mouth. And then afterwards, you need to be absorbed, meaning getting nutrients from food to extract to the body. Now, afterwards, non-nutrients and waste are excreted by defecation. So the first big idea is the gastrointestinal, the digestive system is made up of the intestinal tract and accessory organs. So the gastrointestinal tract is just if you want to imagine your digestive system, it's just one big tube. It goes down to the stomach, then it goes down to the small and large intestine, and then it goes down to the anus. It's just like a big tube. If this is your mouth, like this is a person, this is the mouth, this would be the esophagus and the rest of it. But the idea is your digestive system is just this long tube at the end, and its movement is usually in one direction, although you have a lot of accessory organs to help. Now, the alimentary canal is a hollow, or I should say, the gastrointestinal tract is a hollow muscular tube with a mucus secreting epithelium. So let's look at it. So if we look at it here, the innermost layer is called the mucosa layer. This is an epithelium plus areolar connective tissue. And a little bit above it is called the submucosa, which is filled with dense connective tissue. And then you have two muscle layers, I should say, two muscle layers, smooth muscle, and then you have a serosa layer, which is another epithelium plus areolar tissue layer. So you can kind of have an idea where the serosa layer and mucosa layer are sandwiched. You have a muscle layer, and then you have submucosa, which is dense connective tissue. Now, there's also cases of mesenteries. Basically, mesenteries are membrane that attach over the abdominal organs to the peritoneum. So let's write this down. Mesenteries attach abdominal organs to peritoneum. This helps to hold and to stabilize your organs during their digestive movements. And neurons of the enteric nervous system and autonomic nervous system are found through the digestive tract as well and organized into two plexuses. You can have the submucosal plexus. This contains visceral sensory fibers, parasympathetic, sympathetic postganglionic neurons, whereas the mesenteric plexus contains only enteric nervous system neurons. Now, both of these lead to different functions that we'll discuss later. But first, most parts of the digestive tract, the muscle layer is smooth with two orientations, and they'll have a circular layer and a longitudinal layer. The key exception is one, the oral cavity, pharynx, and upper pharyngeal have skeletal muscle. And two, um, the stomach has a layer of oblique muscle. Three, colon has an incomplete longitudinal layer. That's just three key exceptions. Now, what's also need to note is that visceral smooth muscle, they're connected with gap junction cells, and they are influenced by rhythmically active pace setter cells. They have gap junctions, and they're connected with pace setter cells. So we have a single unit organization. This is similar to the heart. And what this does is it allows for more rhythmic actions, rhythmic movements. And yeah, they spread throughout the muscle layer. There's different functions in the digestive tract that have different specializations. One is propulsion, which is movement. You have digestion, which is food breakdown. You have absorption, which is nutrients entering bloodstream. You have coordination, which is involved in feedback loops and feed forward loops. And you also have protection, keeping pathogens away. So you have these functions. Propulsion refers to the movement of ingested material throughout the digestive tract, and this is known in peristalsis, which is these spreading waves of contraction that propel food little by little in these sort of ballless balls. And yeah, digestion can refer to both physical and chemical. Physical refers to mastication or segmentation that fragment the bolus but don't produce any forward movement. And chemical digestion refers to the turning it into smaller chemicals or enzymes. Secretions of the digestive tract accessory organs enhance both these functions. They can reduce friction, or they can also have digestive enzymes that can help absorption or help digestion. And almost all these secretions are reabsorbed before wastes are excreted. Absorption refers to uptake of nutrients, water, vitamins into bloodstream. And only certain parts of the digestive tract are capable of this function. Now, yeah, mastication, chewing, the function where we have the two types of teeth moving together, chewing teeth. And this leads to physical digestion. The tongue is also another function that helps manipulate food. It conducts physical digestion as well, and also propulsion, because your tongue helps move forward and back. It secretes linguinal lipase, which is for chemical digestion of fats, and it's also helpful for taste. And this is detected with taste cells on the taste buds, which is involved for digestive reflexes. And this can go a lot of long reflexes, which include gag reflex, salivation, and gastric. GSG, gag, salivation, gastric reflexes, which affects taste afferent neurons. Secretion from salivary glands can also salivary glands can also lead to chemical digestion, propulsion, and protection. Saliva contains salivary amylase, antibacterial enzymes, and antibodies, and also this helps lubricate as in aqueous solution since it's 99% water. So what this does is these salivary glands can help maybe chemically digest using salivary amylase for sugars. It can help propulsion by lubricating the digestive tract or the alimentary canal, GI tract, GI tract, and it also has antibacterial enzymes and antibodies for protection. And it's constantly secreted with the digestive tract and enhanced with parasympathetic activity. And you have a salivary reflex, chemoreceptors and mechanoreceptors in the oral cavity. This is a long reflex. I guess, yeah, this is the salivation reflex. You have the gag again on the gastric, the salivation, and then these are integrated into the medulla. So in the tongue, in the oral cavity, you activate chemoreceptors and mechanical receptors. And you just label these two chemoreceptors plus mechanical receptors in oral cavity trigger salivation via long reflexes. And then parasympathetic increases secretion, sympathetic reduces volume of saliva produced, but sympathetic also increases secretions. That's interesting, is that this idea of parasympathetic and sympathetic working together. Swallowing is moving food from the oral cavity, the pharynx, and esophagus. And mainly, what's so important about swallowing, we should say, is that ingested food must move through the inferior portions of the pharynx, and the esophagus must stay out of trachea and esophagus, but avoid trachea. And yeah, the pharynx and the esophagus contain two layers of skeletal fibers. We should say pharynx has skeletal muscle fibers, allowing for some voluntary control. Now, it goes in three phases. One is a buccal phase, and this involves the tongue elevating, pushing against hard palate, like push the bolus, it's hard palate, and then forces into oral pharynx. This is voluntary. And the second, once you're in the pharyngeal phase, this is reflexive, and then bolus presence triggers peristalsis. And we elevate larynx, causing epiglottis to close over trachea. And then soft palate elevates uvula, blocking nasal. So the big idea is you're preventing choking, basically. So after the sort of food is within the bolus is here in the oral pharynx, the uvula and the soft palate. Uvula, soft palate. Muscles of the soft palate elevate the uvula, blocking nasopharynx. And then epiglottis. So laryngeal and hyoid muscles elevate the epiglottis, close over trachea. And this is reflexive. And then lastly, it's repelled into the esophagus. Esophagus, this is also reflexive. So as it moves down the esophagus, a wave of peristalsis begins. So myenteric plexus coordinates contraction down esophagus. And then near the distal end, it relaxes smooth muscle. So I should say stretch receptors, relaxes smooth muscle. This in the lower, I think we should call it lower esophageal sphincter, and it moves into the stomach. And so yeah, this is what would basically happen. The phases of swallowing. Okay, let's talk about the stomach now, since we're finished the swallowing part. The stomach is a muscular pouch. It looks like a little egg like this. And it has four distinct subregions. You first have the cardia. Let's label it. fundus, yeah, the cardia, yeah, the body. Well, you have the, let's say we call this the duodenum. Yeah, the body and the pylorus. So the extra oblique layer gives it strength. So first the order is the stomach is first is longitudinal, and then it's circular, and lastly it's oblique. So LCO for the layers of smooth muscle. And the cardia is responsible for mucus secretions, whereas the fundus and the body are more so digestive secretions. Now, two sphincters control the entrance of the bolus and the chyme. So first is we have on the top is a cardiac sphincter, because it's closer to the heart, and then the pyloric sphincter, because it's part of the pyloric, I guess. These are circular, smooth muscle that can be controlled independently. So circular, smooth muscle. They're contracted by default. So basically they're closed by default. Yeah. So the stomach receives and temporarily stores food and that contributes to digestion. So it has a function of being a storage food. It can do chemical plus physical digestion. This is through churning and the enzymes it has and hormones. And it also has stomach acid, denatures pathogens. Denatures pathogens lead to protection. And this is also coordination via feedback loops, contributes to coordination. It's very expandable because one thing you would find in stomach is because you have these layers of rugae, and rugae is because of wrinkles. Is well, we call them wrinkles basically, and because of this, they allow the stomach to expand if needed. And this is due to mechanosensitive stretch receptors in the wall. The gastromucosa is also a columnar epithelial cell organized into pits and glands. So you have a pit in the top, you have a gland at the bottom, and yeah, pits secrete mucus, which is alkaline, whereas and offers protection against the acidic environment. And the glands have different cells for different sorts of secretions. First, you have parietal cells, which secrete HCl through a variety of transport. Uh, well, they also secrete intrinsic factor and water. Intrinsic factor helps digest vitamin B12, digest, absorb vitamin B12. HCl is produced through essentially the potassium pump. You can get it through carbon anhydrase. And you can, well, there's a lot of ways you can basically produce HCl, but I guess the big way is through primary factor transport. Or I guess in this case, not through primary transport, but rather transport in general. And you also have chief cells. Chief cells secrete pepsinogen. And what pepsinogen is in a low pH environment, it gets converted to pepsin. And this is a zymogen. This cleaves at the pepsin. And pepsin helps digest muscle, digest proteins. Gastrin is also another cell which is secreted by G cells secrete gastrin into the tissue, and not into the gastric lumen. So gastrin is a peptide hormone, and it's increased with the presence of proteins elevated pH. And when there's gastrin acts as a local reflex and it enhances the ability of G cells and glial cells. So essentially, when you're having a lot of protein and the pH is too high, gastrin's going to be like, hey guys, you're going to have to increase your secretion of HCl and pepsinogen to help digest the proteins, which makes a lot of sense. And these are controlled using different, where should I pay for? These secretions are controlled and regulated by different mechanisms. Gastric activity is made by both feed forward and feedback mechanisms, which can be interesting, which can occur in three different parts. So first, we have the cephalic phase, where the CNS stimulates, enhances gastric secretions. This is from the vagus nerve, and it's from the brain. So chemo or mechanical receptors in the oral or pharyngeal, pharynx and esophagus can increase vagal activity on the stomach. So vagus nerve on the stomach. And yeah, this affects the sub. This is a long reflex. And then in the gastric phase, local reflexes, which are neural and hormonal, respond to gastric secretion. This refers to events detected when the bolus or anything reaches part of the digestive system. And this is induced by chemo or mechanical receptors in the stomach wall. And these responses help to enhance secretion of stomach. This surrounds to churning and whatnot, and gastric secretion, and by mixing wave and gastric secretion, this leads to more gastrin gland secretions, frequency, amplitude of churning. And the pyloric sphincter regulates entrance of chyme in the small intestine after gastrin emptying. So after you're done mixing, the pyloric sphincter is open afterwards. And again, this is part of the gastrin phase where they respond to chemo or mechanical receptors. Chemo receptors lead to the opening of the pyloric sphincter. And this is the gastric emptying. And yeah, and then lastly, we have the intestinal phase. And during this phase, the gastrin activity and the rate of gastrin emptying is controlled by reflexes in the duodenum. And they can act to inhibit secretions of the stomach. This slows down the flow of gastrin emptying, slow down emptying, and chyme flux. And the mixing wave and gastrin secretions are inhibited by both intestinal hormones and by neural reflexes. And so these intestinal hormones inhibit the gastric gland secretions. This includes CCK, GIP, and secretin. And in intestinal hormones and enhanced dual inhibit also contractions. So inhibit contractions. Stomach stretching and the release of gastrin are also feed forward short reflexes. So gastrin, feed forward, short reflex accelerates activity down the gastrointestinal tract. So basically, if you have some reflux inside the stomach and with gastrin, it'll lead to freeing up space in the lower in the intestines to induce relaxation of sphincters. So relax pyloric sphincter and free up space for stomach content. Yeah, this increases intestinal motility. Also, yeah, relaxation of the colic sphincter. And that's basically how it is. After you're making it to the, oh wait, I opened the wrong slide deck. So after you made it to the intestines, well, you have the rules of the small and large intestine. Uh, chyme enters intestinal tract, and now you're basically into absorption. Now, the four key points to make is not all can be digested, absorbed. Healthy intestine reabsorbs all volume associated with this, and old intestinal cells are shed into the lumen, or gut bacteria is a large part of fecal matter. So essentially, we can't absorb everything, although it will be helpful, or a healthy intestine absorbs all the volume at least. Old intestinal cells are always shed, and gut bacteria is a large portion. So the lower intestinal tract is three primary organs. First is the small intestine, large intestine, and colon. And there's three sphincters associated. One is a pyloric sphincter, which is from the stomach to the small intestine. And then it's the ileocecal sphincter from the small intestine to the large intestine. And it's the anal sphincter to the rectum. There's also a lot of accessory organs associated, like the liver, the gallbladder, and whatnot. But in addition to size and length, the organization of muscle layers and movement differ between the small and large intestine. The small intestine has a complete longitudinal muscle layer, of which both peristalsis and segmentation occur. But in the large, we have an incomplete. Oh wait, it also only has bands of longitudinal muscle, which means that only peristalsis occur and some compaction. And each of the digestive functions occur in the lower digestive tract, but only the small intestine that has physical and chemical digestion, whereas everything else is basically in all the other organs. Neural and hormonal activity in the small intestine coordinates activity of principal and accessory organs. For example, small intestine, we have the duodenum, and it secretes a lot of hormones, but most importantly, it secretes CCK. And CCK is the duodenal hormone that controls the release of bile and pancreatic juice. So in the presence of fatty or peptide-rich foods in the duodenum, you'll get more CCK, and this goes on to the gallbladder and the duodenal papilla and secretes bile for secretion of bile and pancreatic juices. It also influences the endocrine functions to feed forward. It acts on the endocrine, for the pancreatic juices, it secretes insulin. It also acts on endocrine cells within the pancreas to enhance the secretion of insulin, basically. And yeah, this by allowing for insulin, your rise and preparing for the glucose being absorbed. The small intestine absorbs a wider range of substances, although most nutrient absorption happens in the jejunum. Nutrients, jejunum. It's basically everything from amino acids, vitamins, irons, and whatnot. The large intestine absorbs very few nutrients, but absorbs some vitamins, K, B5, biotin, and especially water. Usually toxins and organic wastes will stay within the large intestine. And you have also malt in regions with large amounts of gut bacteria, such as the ileum, the large intestine. These are important for synthesizing vitamins and chemical digestion. The small intestine also produces more secretions than the large intestine. It has in digestive enzymes, water, and antibacterial substances. 2 liters of fluid per day enters the duodenum from the accessory organs. So I suppose that says something. Whereas the large intestine only secretes mucus. The large intestine is only mucus, I should say. And the organization, because of it, is very different. In the small intestine, you see a lot of villi and microvilli. And you'll see a lot of circular folds for surface area. In the duodenum, you'll see few folds, but duodenum, you'll see few folds with many mucus secreting glands. Jejunum, you'll see many folds and villi. And ileum, you'll see a lot of folds, but also few folds, but a lot of malt. The mucus of small intestine is, yeah, it's a simple columnar epithelium with microvilli. And you also see a lot of mucus secreting cells, goblet cells in villi. And each villi have basically any sort of capillaries and lymphatic vessels. The mucus uh, of the large intestine lack villi, but contain many mucus secreting glands and lymphoid tissue. And the structural is that the mucosa is mainly flat, as mucus secreting glands to reduce friction and protect against harmful bacteria. In the anal canal, the columnar epithelium becomes stratified, so it goes from columnar to stratified squamous for abrasion and dehydration. And yeah, while the rectum is simple columnar, and then stratified squamous, and then it's keratinized stratified squamous. This is for abrasion. And mass movement is within peristalsis, and these are influenced by upward upward reflexes. The rectum can also temporarily store feces. Well, rectum dissentation initiates a process of purification. So this means that once it senses a lot of fecal matter, it's just going to go. The rectum receives fecal matter from the colon and has multiple stretch receptors and motor receptors, and the opening is actually at a dual sphincter. It has an internal smooth muscle and an external skeletal muscle. However, both of them are contracted except at defecation. The defecation reflexes trigger a relaxation of the internal muscle sphincter. So let's talk about the defecation reflex. So inside internal anal sphincter, you'll have a short reflex, which is ENS only. Then you have a long reflex, which is CNS involved via spinal autonomic nervous system neurons. So the external anal sphincter is going to be controlled using a CNS, whereas ANS is going to be controlled, uh, well, I mean, ANS is going to be controlling the external, whereas CNS is going to be controlling the internal anal sphincter, as the internal one is smooth muscle, and then the outside one is skeletal muscle. External one? Oh, my bad, my bad. Both of these, sorry, both of the smooth and the short and the long work on the smooth muscle, and the external skeletal muscle layer is done by somatic motor neurons. This is done from the primary motor cortex M1. This is from voluntary relaxation of it and whatnot, whereas the short reflex and the long reflex is based off the anterior nervous system and CNS involvement. So after this, I guess we're going to talk about chemical digestion and absorption. So the various nutrients we absorb are in the forms of macromolecules, such as carbohydrates, proteins, fats, and nucleic acids. And chemical digestion needs to break down of these macromolecules into their smaller parts. So first we need to talk about the acinar cells of pancreas. They secrete many things that can help us digest stuff. First, they have pancreatic alpha amylase, lipase, nuclease, and proteolytic enzymes. They both help dissolve muscles and stuff, and they're generally secreted as zymogens, only become active in the intestinal lumen. So zymogens are inactive precursors. This is so they don't digest the cell that create them, and they remove part of the protein. They require the removal of part of the protein to express the active site of the enzyme. Remove part to expose active site. And they're mainly activated in the duodenum, like protein-protein interactions. Generally, what happens is enzymes present on duodenal metrocytes convert the zymogen trypsinogen to trypsin in the duodenum. And basically, enzymes in duodenal enterocytes convert this, and trypsin converts zymogens to active. So the big key thing is that an enzyme, it's on the duodenal enterocytes, and trypsin is important. Brush motor enzymes are also intestinal enzymes associated with microvilli of enterocytes. They catalyze the breakdown of small chains of dimers and monomers that are suited for absorption. Some of these include sucrase, catalyzing sucrose, and then lactase for lactose. And they are intestinal enzymes associated with the microvilli. So I'll write that down. Brush border enzymes, intestinal enzymes associated with microvilli enterocytes. The biles contain bile salts that's secreted by the gallbladder that leads to emulsification, which is physical breakdown of fats. And this forms micelles, which allows lipase to access lipid molecules for digestion, for chemical digestion. They are defined as cholic and hepatic molecules, and bile salts are recycled through reabsorption in ileum and transported back to the liver. Other forms of bile, such as bilirubin, are excreted. They can be reabsorbed in the colon, or they're excreted. Absorption of molecules involved basically movement through two membranes, two plasma membranes. So you can first understand like the lumen, the outside environment, and then you have a layer of enterocytes, and then we have interstitial fluid. So absorption has to enter the enterocytes and then the interstitial fluid, or vice versa. It's a two-step process. A remember we have the concepts we introduced in the beginning about permeability. But the big idea is lipids from micelles diffuse directly and are processed by interorganelle organs. This is because so first we have lipids as a class. They can diffuse from micelles across microvilli. And then within the enterocytes, they're going to undergo passive packaging. They're reassembled and add phospholipids. So lipid, lipids assembled with phospholipids into chylomicrons. Chylomicrons. And the chylomicrons are excluded into by the basolateral membrane and transported via lymphatic system into the bloodstream. So yeah, I guess more accurately, it first enters the ENS and then the ISF would be the bloodstream. I'm not drawing that well. They're transported via the lymphatic system into the bloodstream. Uh, Chylomicrons are then released via exocytosis and then enter the lacteals. So why is this done? Well, it's because, well, fats don't dissolve well in I should say, fats are never going to dissolve well in what's it called? In the bloodstream, because the bloodstream is made out of water. Fats are non-polar, whereas water is polar. Because you can't have the rule of like dissolving. There has to be some way of passing it through and also dissolving in the bloodstream. And first of all, well, if you want to, I should say, if you were to first know, that's why they undergo the packaging into these chylomicrons that can pass through let me see, that can enter, that are passed through in enterocytes because they can easily enter the enterocytes, but then the chylomicrons be packaged then too. After exocytosis, it's just sent to the what's it called, the lacteals of villi they're transported via the lymphatic system through the lacteals and then before entering the bloodstream. And the monosaccharides are absorbed via the lumen through either facilitated transport or co-transport with sodium ions. So sugars, we should say, they have a facilitated diffusion using glucose glut transporters, or they have some sort of NAK ATPase pump that basically just help that, yeah, is active transport, primary active transport, moving it in. Monosaccharides enter the intestinal, I should say, move into the interstitial fluid this way. Whereas it's diffused into well, it's either diffused or they move with sodium into the enterocytes from the lumen into the enterocytes, and then later they either get diffused or another ATPase pump inside. And monosaccharides enter the intestinal capillaries that flow into the portal vein system and enter the capillaries of the villus, and this goes to the portal veins, and this eventually leads to the liver. At the liver, glucose is released in the blood or stored as glycogen. So glycogen or to blood. So amino acids are a combination of co-transport and acylate diffusion. It's similar to sugars, whereas it first uses Na co-transporters to enter in the intersects, and then it's likely just co-transported inside into the bloodstream and stuff. And they also pass through the portal veins to the liver. So I think it's relatively similar to carbohydrates. The lower digestive tract is also responsible for reabsorbing water, minerals, and vitamins. The vitamins could be different in terms of structure, whereas small or large, hydrophobic, hydrophilic, and whatnot. And vitamins are also, you can't make them in your body on its own for the most part. So it's definitely something that is key for your body to maintain these normal functions. Uh, it also absorbs minerals like ions in terms of primary and active transport and is regulated by several hormones. Ions, like you can either, well, ions can either diffuse or bulk transport, or paracellular transport. Uh, bulk transport or paracellular, this is one way. It can also have an ion, you can also have an ion channel, or it can have like a co-transport thing going on. And this is regulated, independently regulated. So this depends on the situation, depends on where in the intestinal tract. But some examples include aldosterone, which increases salt uptake. I believe it's in the kidneys, and PTH and calcitriol increases CA uptake in the bloodstream. Water also crosses through the leaky epithelium through osmosis via paracellular transport. So water is through paracellular transport via osmosis. And they have this net movement of solvents. Movement of solvents makes osmotic gradient. And this helps travel. Imagine if the solids are moving in one way, then water can follow. Because there will be this gradient being made where if there's salts on the other end, then there's going to be an imbalance. And the mechanism for vitamin absorption depends on their size and composition. So let's say it's water soluble, so hydrophilic, and let's say it's small. Then you just have facilitated diffusion using facilitated diffusion. But let's say it's lipid soluble, and we're going to get transport with micelles and chylomicrons. And let's say it's water soluble but large. Then it requires endocytosis and needs a protein cofactor. And in the case of one for B12, that would be intrinsic factor. So if it's small, you can just small and charged, well, you can just enter through a facilitated diffusion. If it's lipid-based, you need chylomicrons or micelles to help move it. And if it's charged but big, then you need a protein cofactor, and it requires endocytosis. Now, most everything that we see enters the portal vein system and passes through the liver before general circulation. And patterns of metabolic activity occur generally can be divided through two general patterns. One could be absorptive and post-absorptive. Absorptive, you can think of it as anabolic, so breaking stuff down. And the post-absorptive is catabolic. Sorry, I'm going to double check this actually. Oh no, catabolic is breaking stuff down, and anabolic is making bigger things, but it's like A to A and PA to whatever. And then in absorptive system, you need insulin for uptake, and then glucagon the post-absorptive for building up. So again, catabolic is breaking down, whereas anabolic is building up. So in the absorptive state, you're having built up energy stores as catabolic, whereas post-absorptive, you're retrieving energy stores retrieved. And then absorptive, your increased liver glucose, whereas in the post-absorptive is liver release glucose. And the absorptive glucose is equal to ATP. Post-absorptive, you're using fatty acids as ATP. Fatty acids are good for long-term energy, remember. The absorptive stage occurs usually when food is absorptive, begins to absorb, and the primary hormone is insulin, which allows for storage of lipids and use of blood glucose as ATP. Post-absorptive stage, you have glucagon, epinephrine, and glucocorticoids, which includes gluconeogenesis, lipolysis, and use of fatty acids for ATP. These are big things. And the last thing we'll talk about, what you call it, is we're going to talk about the liver. So liver has many, many, many roles that help in digestion and also aside from digestion. Sorry, give me a sec. I need to find a slide deck. Okay, so remember how we have the absorptive and post-absorptive stages? Uh, so the liver, the design place of the liver is a control hub for control of many metabolic functions. So in the absorptive stage, well, anyway, the liver is anchored in the right superior portion of the abdominal cavities by the peritoneum and the mesenteries, and is attached to the diaphragm by the peritoneum and stomach, anchored to the stomach by the mesentery. So attached to diaphragm by peritoneum, attached to the stomach by mesentery. And it also has a lot of hepatic ligaments. Folds of the mesentery divide into hepatic ligaments into lobes. You also have specific large ducts and vessels. You have a hepatic vein, hepatic portal vein, and whatnot. But yeah, I mean, the main idea is, if you want to, let's draw it out actually. You have a liver like this. You're going to have hepatic vein at the top. Hepatic vein. You're going to have a gallbladder here, which is going to be very noticeable. You have a bile duct. And then here you're going to have the hepatic portal vein. You're going to have hepatic artery. And this complex we have is called the porta hepatis, which is the door to the liver. And the hepatic portal vein collects blood from many organs within the abdominal cavity into the liver. And these organs are involved in many functions, including absorption, digestive, coordination, metabolic regulation, and blood filtering and recycling. And the blood of these organs is diverted towards the liver before it actually reaches the circulatory system. The liver is vascularized, contains hepatocytes organized in lobes. The capillaries have a sinusoidal endothelium. This leads to basically because the liver has a lot of functions in producing, recycling proteins, plasma proteins. It needs ways for things to move out, and there are also specialized for a a resident population of stellate macrophages for their own immune function. There's many functions of the liver. I'll list out some of them. First is synthesizing and secreting bile, storing glucose and lipids, maintaining normal concentrations of glucose, amino acids, fatty acids, and bloodstream, synthesizing nutrient types, synthesizing and releasing cholesterol, inactivating toxins, storing iron, storing fat, soluble vitamins. So first, the A1 liver secretes bile, important for both digestion and excretion. And bile contains bile salts, which is important for fat digestion and absorption, and it also contains bilirubin, toxic waste product, AIM. Uh, and the other function two, it has functions in glucose processing, storage in absorptive stage. So it first stores glucose as glycogen, but it also uses glucose to make new triglycerides. Triglycerides, which are like fats and stuff. And function three, liver uses as a store for minerals and vitamins. So especially for minerals, you're going to see vitamins A and well, sorry, vitamins, you're going to see A and B12, and you have metal ions such as Cu and Fe. It maintains homeostasis by storing these. A4 is used to gluconeogenesis during the post-absorptive stage, converts glycogen into glucose, and synthesizes new glucose from get amino acids and glycerol, converts amino acids and glycerol into glucose as well. It's also helpful for detoxification of alcohol, drugs, and toxins. They have enzymes and cofactors that allows for digestion. One is actually alcohol dehydrogenase and alcohol dehydrogenase and ALDH aldehyde dehydrogenase, and it breaks down alcohol to ATP generated substrates. Biochemical pathways in the liver are complex and they interact. Sometimes one it can interfere with others. Processes can compete because they need the same cofactors, or they can downregulate each other. For example, ethanol metabolism leads to signaling that increases fatty acid synthesis, but inhibits gluconeogenesis. So let's say in the liver you have ethanol breakdown, and this can support fatty acid synthesis. This can also inhibit glucose synthesis. And they have also functions that are not directly related to the metabolism. You can, as I mentioned before, synthesize plasma proteins. You can also synthesize clotting factors. You can recycle old red blood cells. You can store blood, and at rest, it stores around 25% of the cardiac output. You can also absorb hormones and immunoglobulins. And lastly, absorb lipid soluble drugs. So A5, I guess, you can detoxify alcohol drugs as well. Beyond alcohol, uh, and lead to excretion. For example, hepatocytes, those sites inactivate Tylenol for excretion. Another function C, soluble proteins are synthesized in hepatocytes. Hepatocytes and join the bloodstream through sinusoidal endothelium to bloodstream. This includes albumin, globulin, fibrinogen, and others. This includes clotting factors, most clotting factors, complement proteins, and transport proteins. Function D, it can recycle red blood cells. Remember the stellate macrophages? Stellate macrophages? It phagocytose red blood cells, recycle content, especially the iron in them. And it's also a secondary endocrine organ. It secretes at least 4 hormones or 4 prohormones. For example, it secretes in response to GH, growth hormone from the anterior pituitary. It secretes somatotropins for growth. You can also secrete angiotensin, is secreted by the liver. Oh, the liver produces angiotensinogen. It's converted to angiotensin when blood pressure is low in the liver. There's also thrombopoietin in the liver for platelet formation, and also hepcidin for iron homeostasis. And lastly, yes, I'll use this top page. The liver contributes to activation of steroid hormones, activation or inactivation. Steroid hormones. So lipid derivatives, often broken down. They can be broken down in liver, but also liver to cholesterol synthesis processes cholesterol derivatives such as steroid hormones, and it also synthesizes hormone calcitriol from vitamin D, which enters the digestive tract for greater what's it called? Calcium absorption. And when they're damaged, you can first accumulate fat. Liver cells damaged, they first become fat, and then they turn into collagenous scar tissue. And yeah, and this is due to a lot of drinking, one of the causes at least, and this can lead to cirrhosis, end stage liver disease, where scar tissue has replaced functional tissue. And this is really bad. This can lead to a lot of damage and stuff. So I guess this was basically the liver and all these cases. And I hope you enjoyed. We have one more lecture and one more outcome out, which is just the digestive system, or I mean the urinary system, and then we'll be done for anatomy and physiology. Thanks for listening.